Hello students, today we will be studying about the various types of milk. Now this various types of milk can be categorized based on the different biochemical activities that is happening in milk because of the microorganisms or the various physical changes that is done to the milk so that to enhance the flavor or to increase the shelf life of the milk. So based on these two categories there are various types of milk. We will study that in today's class. Now as you know that milk is a complete food, there are milk when it is secreted by the animal, it is free of microorganisms, but as it is rich in nutrients, when it comes out and when it comes in contact with the various sources, microorganisms reach the milk and the milk can get contaminated. Okay. Now when the microorganisms reach to the milk or reach into the milk through these various sources, these activity of microorganisms will bring about certain biochemical changes in milk because of the metabolites that they release out into the milk. Now this activity of these microorganisms will either be beneficial, which will probably say enhance the flavor of milk or will enhance the shelf life of milk, or it could result in the spoilage of milk. Okay, because all this happens because of food is rich in nutrients and microorganisms easily survive in this condition. So as I have told you, milk when freshly drawn, it is free of microorganisms. There are biochemical changes which takes place with the help of microorganisms, which will change the flavor. It can deteriorate the flavor or it can enhance the flavor. There is a change in appearance and of course the change in texture. Either it can enhance or it can spoil the milk. Now the various biochemical activities or biochemical changes that happens in milk because of these microorganisms are souring, proteolytic activity, gassy fermentation, lactic acid production and lipolysis. Let us see each one in detail. What is souring? Now milk, when you keep it you keep it out at room temperature for about 4 to 5 hours, what happens is these microorganisms will start multiplying. They will start consuming the nutrients that is present in milk and they start releasing out the acid in the milk. Okay, as the milk becomes acidic, what will happen is the coagulation of the casein protein will start happen and the whey will start separating out. Now when will this happen? The pH, when the pH of the milk reaches about 4.6 to 4.7, at this particular pH the milk will start curdling and the acidity percentage, that is 0.5 to 0.65, within this range, if the percentage of acidity reaches, then the milk will start curdling or souring. Now this could happen because I am deliberately adding these microorganisms into the milk, which is done in controlled conditions like your lactobacilli, leuconostoc, streptococcus thermophilus, streptococcus liquinifaciens, bacillus coagulans and coliforms. Basically bacillus coagulans and coliforms are the ones which will result in the spoilage of the milk, whereas all the other organisms mentioned will bring about a beneficial change in the milk. Okay, now all these beneficial changes in milk should happen only under controlled conditions, only under controlled temperature and also under a certain number of microorganisms. Of course the number should not exceed a particular level. Okay, how do you control? What do you mean by controlling the conditions? Again, by maintaining hygienic conditions, maintaining hygienic practices, so that only that required organism is growing and in that specific number it is growing. The milk has to be stored in appropriate conditions even when you are allowing the organisms to grow. Also, once the souring process is over, you must not allow the organisms to overgrow there. Okay, and also proper pasteurization. Next one is sweet curdling. Now what is the meaning of the sweet curdling? Curdling, we all know that when the acidity of the milk increases, the casein proteins will start coagulating. Sweet curdling is and you get kind of lactic acid production, and that lactic acid taste will be there. Sweet curdling is, you know that milk is sweet in taste because it has a sweetish flavor because of the lactose sugar that is present in milk. Only when the milk starts acidifying, then the curdling will start. But here what happens is the milk, the sugar is not broken down. by the microorganisms or by any other process, the sugar remains in the milk, but the casein protein starts coagulating. This happens because of certain microorganisms, or also because of certain enzymes like renin or renin like enzymes, or because of the microorganisms like streptococcus lichenifaciens. Now what do is they, instead of breaking down sugar, they will break down proteins, or they will break down other components in the milk, and will result in the coagulation of the milk. So the sweet taste remains in the milk. That's why it results in sweet curdling. Okay. Now where do you see this? Generally it is more prevalent in summer. Okay, so this is about your sweet curdling. Next one is your proteolytic activity. Proteolytic activity, here it is breaking down of proteins by the microorganisms. Generally when the proteins break down by the microorganisms, what happens is there is a kind of bitterish flavor that is left behind. Okay, the milk becomes bitterish in flavor. So generally this is not an acceptable flavor, but so in certain conditions this bitterish flavor is taken up, probably like in certain milk products like your cheese. Okay, this bitterish flavor is required when the peptides, when the proteins are broken down to give rise to peptides, this bitterish flavor is obtained. Okay, now it also gives a kind of unpleasant flavor, but then this does not go to extreme. Slight bitterish flavor is accepted or is used in the processing of certain kind of cheese. Example of certain proteolytic bacteria are, most of them are psychotrophic in nature, like your Pseudomonas species, Achromobacter, Aeromonas, Enterobacter and Lab. Enterobacter, Aeromonas are these organisms who are not done in controlled conditions. They are they result generally in the spoilage of milk, but whereas the others are added. Even Pseudomonas are it's done not in controlled conditions but as a source of spoilage, but then the all the others, lactic acid bacteria, this is an organism that is added so that you get that flavor, that bitterish flavor. Okay, so this is of proteolytic activity, generally used in the curing of cheese, in the cheese industry. Next one is your gassy fermentation. Here generally you may require certain amount. Here what happens is microorganisms, they break down this sugar and they release out acid and gas as the end product. Not all microorganisms do both. Most of the microorganisms, they release out acid and gas as the end product. Some of them, they release out only acid as the end product. So we select certain organisms who will release out acid and gas as the end product after breaking down the sugar. Okay, carbon dioxide, generally carbon dioxide is released out. Now when this kind of condition happens, what happens to the consistency of milk? It becomes frothy in nature. Okay, why? Because of the excessive accumulation of this gas, it becomes frothy in nature, and the milk becomes little thick. Generally you see this, why do you require, is it added deliberately to enhance the flavor, or it results in the spoilage? Most of the time it is spoilage, but sometimes it is added in controlled conditions. Why? To get certain milk products, like for your cheese. In some of the cheese you might have seen that there are eyes. Okay, there are holes in the cheese. Now these holes is because of this carbon dioxide that gets escaped out, and hence that is the reason you get that particular texture or that particular shape or that particular those eyes in the cheese. So in order to get those eyes in the cheese, carbon dioxide is required. That is where you utilize these microorganisms. What are those organisms? Organisms like yeast is added, candida is added, and pseudotropicalis is added, torulopsis is added, coliforms are not added deliberately, but again it is a contaminant that will result in the change in the milk. Clostridium, again this is an organism which is a contaminant. Also you can find this kind of change or undesirable change in cans. You might find that these organisms will be growing in the cans where there is absence of oxygen, and hence they happily grow and they release out carbon dioxide. You find that these cans are swollen, these tetra packs are swollen, which means there is release of gas from the microorganism, and hence the shape of the can is changed. Okay, how do they reach the milk? From the feed, from the manure, from the soil, etc. Sometimes it is deliberately added, as I told you, in the cheese production. The next one is lactic acid production. This is the one which generally is used in many of the curdling of the milk, wherein I want to convert the milk, that is a milk sugar, to lactic acid. There can be two types. One is your homolactic acid fermentation, and one more is your heterolactic. Homolactic acid fermentation, only lactic acid is given out as an end product by the microorganisms, and this is done in controlled conditions, accepted. And heterolactic acid fermentation is, along with lactic acid, there is carbon dioxide that is released, there is butyric acid that is released, there is acetic acid, there is, all these all things are also released, which may not be a desirable change. It can result in the spoilage of milk. Okay, so fermentations like your lactic acid, citric acid, alcohol, propionic acid, all these components will be released out as a result of heterolactic acid fermentation. Now, what kind of pathway that these organisms follow to break down lactose to lactic acid? That is, first lactose is broken down to glucose, and then this glucose is broken down to lactic acid and other components like acetic acid, etc. Which is that pathway? Phosphobeta galactosidase pathway or hexose diphosphate pathway. So these are the different pathways that the organism follows for production of lactic acid. Okay, now what are the organisms? That is Lactococcus lactis, Lactococcus cremoris, and Lactobacillus. These are the microorganisms. Now the kind of milk or the type of milk, based on consistency, there can be different types. So like your condensed milk, your powdered milk or dried milk, and then the liquid milk. Okay, the condensed milk. What is condensed milk? Condensed milk, all this is based on your consistency. Physical changes happen to the milk, but it is not due to the activity of microorganisms. Okay, probably I am adding certain ingredients to the milk, or I am removing certain ingredients to the milk to bring about desirable physical changes in the milk without the interference of microorganisms. So these are non-biochemical changes in the milk. So what is condensed milk? It is the evaporated whole milk, and condensed milk can be of two types. One is unsweetened condensed milk and the sweetened condensed milk. Now these condensed milk can be either prepared from cow's milk or buffalo's milk, or it could be combination. Most of the time it is a combination of milk. Now in condensed milk, what are the things that is added? Generally calcium chloride is added, citric acid, sodium citrate, and sodium salts are added. It is made sure that the fat content is more here. Again, it is a thick milk. Okay, water content is eliminated, and the fat should not be less than 8%, and the milk solids, you know what is the meaning of milk solids? So total solids, so these solids should not be less than 26%. In sweetened condensed milk, what are the ingredients that is added? All these above ingredients like your calcium chloride, citric acid, sodium citrate, sodium salts, plus your lactose. Okay, and here of course the fat content is around 8 to 9%, but the milk solids will definitely increase because you're adding another ingredient, that is lactose. So your milk fat, your total solids should not be less than 31%. So this is about your condensed milk. Now how do you prepare this condensed milk? It is important as to what kind of milk you select. Basically, as I told you, it's the buffalo milk and the cow's milk that is selected, and this milk is homogenized properly, and then it is set for filtration. Now why? Because unwanted impurities could be there, hence which is maintained at 38 to 40 degrees centigrade, and it is filtered so that other unwanted impurities, physical impurities, are removed. Later it is set for standardization. Now standardization meaning we know that the milk should contain this much amount of fat, the milk should contain this much of sugar, the milk should contain these nutrients, these amino acids, these proteins, or this vitamins. So made sure that all these ingredients are there in that specified amount. So that's called as standardization. And once it's the standardization is there, then the preheating process starts, or it is also called as a forewarming, which is maintained at 45 to 48 degree centigrade. Once at this condition, when the forewarming is done, sugar is added at the required quantity, if it is required. If sugar is required, that is, for the sweetened condensed milk, sugar is added, and for unsweetened, sugar is not added. Now then the condensation process happens in the ratio 2:5:1 for all the ingredients that is mentioned, and once all the ingredients are added and it is there in the required proportion, it has to be made sure that all these ingredients are evenly distributed throughout the milk, and hence homogenization is done. Once the homogenization process is done, the milk is cooled and if required crystallized, and after it is cooled, which is kept at 5 degrees centigrade. I mean to say, milk was kept at 45 to 48 degrees centigrade. Slowly the milk is brought down to room temperature, and then it is set for packaging. It is sent for packaging and dispatch, and then it is stored at 10 degrees centigrade as long as it is stored. 5 to 10 degree centigrade. The next one is the skimmed milk. Now what is this skimmed milk? Skimmed milk is wherein the most of the fat content of the milk is eliminated, or it is removed. Probably a person requires the all the nutritional value of milk, but except cannot take in the fat content, and hence the fat content is removed, so that the person gets all the nutrients except the fat. Here generally cow or buffalo milk is taken. Generally it is a cow's milk that is taken. What are the additions that is added? Calcium chloride, citric acid, sodium citrate are added. Here it is made sure that the fat is not more than 1.5%, and acidity definitely should not be more than 1.5%. How to prepare the skimmed milk? It is the same process as how we prepare the condensed milk, except for that the fat is not increased and the water is not evaporated. Moving on to the next one is your milk powder or the dried milk. Here the water is completely eliminated out. Made sure that moisture is not more than 5%. Now why do you do this? Because if you have the normal milk, that is if you have the liquid milk, it gets spoiled within a day or two. Why? Because moisture content is high there, and microorganisms require moisture to carry out their metabolic activities. But if I remove the water content, this milk powder could be stored for a very long duration of time, and hence the shelf life of the milk increases. When I want to use the milk, I'll just take in the milk powder and mix it in water and consume it, or as per the requirement. So it is generally prepared by taking in the whole milk and then evaporating it by various processes, evaporating the water content and retaining the solid milk solids, the total solids. It has to be made sure that the moisture content is not more than 5%, and this kind of milk is called as a dried milk or the dried whole milk, or it is also called as whole milk powder. The liquid milk is called as whole milk otherwise. Okay, and generally it is prepared from defatted milk. As I told you, the fat should have been removed, so it's generally the cow's milk rather than the buffalo milk, or it is the skimmed milk which is utilized here. So these are the various types of milk. One is the milk that is obtained by the biochemical activities of milk, that is the based on the biochemical types of so that is called as a biochemical changes that is seen in the milk because of the activity of microorganisms. The next one is the physical changes in the milk because of addition of certain physical ingredients in the milk without the activity of microorganisms. Thank you, students.